Hello, my name is Micael Roldan, and today I will present my research entitled Differences in Sugar Response Threshold and Sugar Satiety Levels in Pollen and Nectar Foraging Bees, Apis mellifera. Social insects such as ants, termites, and some bees exhibit division of labor, meaning that different individuals specialize in performing specific tasks to enhance the colony's efficiency. The division of labor between pollen and nectar foraging bees is a well-studied case that demonstrated how intended individuals' differences in sensitivity to sugars affect preferences for collecting one resource or the other. These differences in sugar sensitivity are estimated by bees' sugar response thresholds, the lowest sugar concentration in a solution that triggers the extension of proboscis. These sugar response thresholds are dynamic and adjust according to factors such as forager motivation or nutritional state and the quality of previously experienced food. Our goal is to determine whether sugar response thresholds of pollen and nectar foragers are related to their previous experiences or are inherent to each group. Additionally, we will assess whether differences in sugar response thresholds correlate with bees' sugar satiety, estimated through crowd load. To address our first objective, we train bees to visit foraging stations equipped with a 20% weight in weight sugar solution feeder and a pollen feeder, with a manually captured 70 bees of each group as they arrived at the station, ensuring that they were highly motivated to collect resources. Half of the bees in both groups were evaluated 30 minutes after capture. The remaining bees were placed in a pot with ad-libitum food to normalize factors that could influence their sucrose responsiveness. After 24 hours of confinement, we assessed sugar response threshold of this second group. To estimate sugar response thresholds, the bees were cold anesthetized and restrained. We then applied a protocol in which the bees' antennae were touched with a stick dipped in ascending sugar concentration, with intermittent presentation of water to prevent responses driven by thirst. To correlate sugar response threshold with sucrose satiety, we conduct a second experiment. We repeat the capture and confinement procedure and then dissected the bees under a microscope to extract and weigh the crop content, serving as a measure of the bees' satiety level. All data were analyzed in our environment. In Figure 1, the x-axis shows the different sugar concentration and the y-axis shows the probability of a response. The graph reveals that nectar foragers display lower sugar response threshold than pollen foragers, evidenced by their higher probability of responding to sucrose solutions at lower concentration. Normalization had an effect on sugar response threshold. However, we found no interaction between forager type and normalization. In Figure 2, we can observe the average crop weight of both groups. It is evident that the crop of pollen foragers were more loaded than those of nectar foragers, both before and after normalization, with the differences being statistically significant. This means that pollen foragers will have a higher sugar satiety level than nectar foragers. Our results suggest that the differences in sugar response thresholds between the pollen and nectar foragers are not due to their prior experiences or motivational state during resource collection, but are inherent to each group of foragers. In line with previous evidence showing that bees increase sugar response thresholds when satiated with carbohydrate, the differences in sugar response thresholds could be explained by the differences in satiety levels, with the pollen foragers showing a higher food supply in their crops. Differences in sugar response thresholds may provide an adaptive advantage by maintaining food separation and ensuring that both foraging tasks are performed efficiently for the colony. Thanks for listening.